We're going to be talking about the origin of life. If you were here for the video we just watched, that gave you one taste of what the cutting edge is of the science of the origin of life. We're going to hear a very different story now from Bruce Damer. He is a researcher from the University of California, Santa Cruz, and he is a polymath in so many other ways. I met him last week, and just kind of over a chance evening of talking over all matters, scientific, spiritual, about space, about the future, about the past, about everything, we decided that, hey, why not? Let's throw a talk here. And I think that this synthesis of all these different fields that he studied and all the angles that he takes it from really provides a holistic angle on this that I think gives a real depth to this work. I'm really excited that in particular we have some actual experimental both evidence and hypotheses that can be tested, and he's going to talk about all of that now. So, everybody, Bruce.
Thank you, Eugene, and thank you, Kaleidoscope. It's such a colorful, beautiful place here. It is a true kaleidoscope, and your faces are part of that. That beautiful mixture, texture. I see some old friends here. So what I'm going to describe to you is actually pretty new. So Eric's talk is from 2015. He's our colleague at ELSI. And for about 30 years, so our field, actually I'll just go right. Take a look at this first. This might need some volume. I'm going to dive, I'll go back. I was just in New Zealand, and we're on a tear. We have laboratory evidence of the kinds of things Eric merely waves his hands about. It shows these citric acid cycles and those kinds of things. We're now in the full scale emergence into laboratory, empirical, and field testing of this hypothesis, which is an origin of life on land in cycling hot spring pools, which is where Charles Darwin originally intuited that life can start in a warm little pond somewhere. So this is actually returning our field after 150 years to the warm little pond. So what I'm going to do is show you now. I was face down in a hot little pool about 3 weeks ago. Let's see if this stuff comes up.
It's just a case of weird science. Leading American scientist Dr. Bruce Damer is at what to do as Hell's Gate, conducting research trials aiming to answer the age-old question, how did we get here?
It's very exciting. It's 21st century science, revolutionary science to watch. If we can find this cycle, it's the engine of creation effectively. Also that we came not from an individual, not from competing individuals, but from a network, a collaborative community was at the taproot of the tree of life.
Dr. Damer's research could turn previous origins of life science on its head. He's testing whether hot springs on land may have been the starting point for life itself. Dr. Damer's searching for RNA, a nucleic acid, and the basis of all living cells.
So what we think is that RNA could have been the first polymer that life synthesized first at random in a process like this, and then picked through selection, through these cycling protocells, which we're doing here in the hot spring, picked ones that worked.
He's also hoping to conduct research into the formation of a molecular structure much closer to home.
It's possible also that DNA could have formed in a condition like this, self-assembled. So the little double helical polymers self-assembling just like we're doing with the RNA here, and that DNA may have been around the same time as RNA, and then they could interact, and the DNA acts as a data store for the RNA, which reads the data store and then makes all the proteins that make life.
For years, scientists had accepted the theory that life began in hydrothermal vents in the ocean. Charles Darwin once speculated that biological molecules might form in a warm little pond, and Dr. Damer believes this to be much closer to the truth.
If we can show to our colleagues and to the world that we can self-assemble an important biopolymer of life called RNA in a hot spring pool that's cycling, in the conditions that would have been around in the early Earth on these big volcanic islands. Our colleagues are really going to seriously look at doing more experiments. And it'll create a whole movement, a whole sort of paradigm shift in origin of life thinking, back to Darwin's warm little pond, only now it's a cycling hot little pool.
Jade McLeod, Local Focus.
So that's the new work, and I'll show you this is 2 days ago. This is what our gel showed in the lab. So I don't know if you can see this on my phone. I just switched it to
Go pass it around, I guess.
Well, I'll kind of word it. These bands, these streaks here are RNA, although the two monomers all the way up to about 200, 250 base pairs long, produced in those vials that you just saw. And this is such high productivity compared to our laboratory work that we're like beating the lab. So we have these pristine conditions in the lab, put an acidic buffer in, put AMP and UMP, put the basically the molecules of amphiphiles of membranes, and we've been doing this for 10 years. And when we did it in the field, it's like rockingly more productive. Because there's something going on. There's ionic solutes in there. There's something that's making this reaction just rock, like super fast. So we'll go get right into that. So as you saw, probably those of you who were here for Eric's talk, Darwin in 1871 intuited that if, and what a big if, right? If we could conceive in some warm little ponds all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. He got it all right here. Present that a protein compound, and the Victorians did not know about nucleic acids. They knew about proteins. So they sort of assumed that protein was important. But the protein compound was chemically formed, ready to undergo still more complex changes. This is the key phrase that people don't really repeat. That actually is where Darwin nailed it. You have to grow a long stringy polymer, you have to form it, and then you have to reform it and reform it and reform it, elongate it and make it more complex. It's called away from equilibrium system. But life starts with polymers. And he nailed it in 1871. This is it. This is the whole story. And we're going to show you how we've now extended from Darwin. We've gone back to Darwin and we've now pushed forward in 21st century science. So as probably Eric also described, you had these idea of soups, JBS Haldane and Oparin in Russia, talking about you need to have things put together so that more complex things form. That was sort of the primordial soup approach. And then you had Miller and Urey, this is Stan Miller here, with the spark chamber making amino acids in a prebiotic plausible atmosphere, which turned out not to be actually necessary, as we'll show you later. And then 1977, Corliss predicted and discovered the hypothermal vents. And in 1982 suggested that life might start there because there's a lot of life there now. And so the field actually went down this rabbit hole for 30 years. And the chemists never liked this because if you took laboratory solutions that were prebiotically plausible, some monomers, some membranes that would fall from space, because your membranous material is constantly being delivered by asteroidal input, and you built a simulator, you went to this environment and attempted to do the chemistry, it would just blow the system apart. It would dissipate it everywhere, you couldn't form polymers. The little membranous compartments that you might be able to form in the lab would collapse and crystallize. So this is an extremophile environment. Life probably needed hundreds of millions of years or a billion years or more to adapt to this setting. It's an incredibly harsh environment. So the geochemists love it because it's hot water rock interaction. The physicists love it because, well, there's these things, there's batteries and things that physicists understand, but it actually doesn't have anything to do with the origin of life. It's a chemical garden forming environment that's very, very harsh on the living world. So you can counter me along the way with any challenges I'll take it. So this work was percolating along for 20 years with Dave Deemer at the Deemer lab at UC Santa Cruz. I met him in 2009 and we started to collaborate. And then, by fortuitous chance,
the Dimer-Damer-Dimer?
the Dimer-Damer, it's called the Dimer-Damer hypothesis. That's confusing because his name's a one-letter mutation. Mine. Anyway, and it turns out that our relatives were in London at the same time in the 1550s. Found out there may have been some crossbreeding. Many of you. Hi. Good to see you. So the crossbreeding aside, in 2015 we met a fellow named Martin J. van Kranendonk, a Dutch name which means something about geese delivering babies or something like that. But he's a Canadian like me. He's one of the, what you call the big glasses geologists. Like, he's got glasses like yours. They're intense. They walk like 1 million miles and they sample massive environments and they're high energy. And he had more energy than a kangaroo, I'll tell you. And he was in charge of the Pilbara, the northwest of Australia, the geological region, which is the oldest piece of Earth's crust that's intact, that was well preserved. And they found in the Pilbara, they found this stuff. So this is late one night, the same month that Dave and I were coming up with our hypothesis, they found geyserite in the Pilbara. And you can see these dark light bands. That's basically a mineral put down by splattering water out of a freshwater hot spring. And within the geyserite, these terracet fabrics here, which are indicative of microbial mat community. And even more stunning, these little sort of vacuoles here, which are interpreted as preserved air bubbles, oxygen bubbles, which you find inside what's called EPS, which glues microbial colonies together today. And it's the oxygen that's popping off of the microbial mat, the sort of slimy stuff. And these are in silica preserved air bubbles. This is the oldest evidence for metabolism on Earth. But this is the oldest evidence for life on land. It's in a hot spring on the surface. And the oldest evidence for metabolic activity. 3.48 billion years old. And I have a piece of it here. This is what it looks like. This is a spalling that I took off of when I was there. And that literally, the little sort of dark white specks in there are a branching stromatolite, and on the top and bottom of the sample is the geyserite. So this is definitive evidence for life on land as far back as we can go, and a very vibrant community in a hot spring. So it's like a sign pointing back. So the mythology that the continents were a dead place and that somehow life crawled out on land is long gone. You know, I found this in 2016. This is actually a new stromatolite. This is brittle red balls here. That's stromatolitic material balled up inside barite, in a barite mineral. We haven't even published anything on this yet. I just broke a slab open in this new locality and boom, more stromatolites everywhere. It's fantastic. And then in here, this is something we've named Precious, because when I took it to New Zealand, it became precious, and it was going to go back and be presented to Gollum at a studio. They gave me Glamdring to hold, because Sir Richard Taylor thought I looked like Gandalf the Grey. And then when we were walking, they gave us a tour of the whole studio, and I gave a talk there, and I presented Precious to Gollum, who wasn't interested, and he was in the middle of eating a fish. And then it was just a blast. I got to hold Glamdring, the whole thing, but actually Sir Richard Taylor grabbed this thing, sent it downstairs. They 3D printed it. And their artists are actually painting it exactly, because their artists paint all this fantastic stuff for all the movies you watched, like Blade Runner and Lord of the Rings and stuff. So that's what happened. All of this is in the news again, because last summer, in August, Scientific American's cover was this new hypothesis. And you saw me holding this up in the video. And they just published it again in this special issue. So on newsstands, if people still buy things on newsstands. And it's a real honoring thing, because we're the lead article in this. So this is, a dozen revolutionary sciences for the 21st century. So there's the entire hypothesis. This took 2 years to come up with this diagram with about 50 colleagues for an origin of life scenario that's testable and is attached to space, it's attached to landscapes, it's attached to chemistry and thermodynamics and combinatorics. So
You want to pass it around?
yeah, actually, it's just here. You want to pass it around? Please feel free.
By the way, the evidence of metabolism you bring up, I'm curious for a little more info on that.
That's these spherical silica-filled contusions everywhere inside this material. It's really the only way to preserve that is to start with a sphere. And the silica is an exquisite preserver of life. And I'm on Mars 2020 landing site selection, and we're trying to get NASA to go to a silica hot spring so we can look for things like this and like that.
Okay, and so the spheres of silica are produced by organisms from some process?
So actually, we'll get to that later.
Okay.
That comes with the Yellowstone story. So here we have it, the continuity in the rock record. So you have hot spring evidence. You have, this is a subsurface vein evidence. I can even hand, do you want to hand Precious around? that's Precious is lakeshore. So what you're holding in your hand is a 2.7 billion year old lakeshore microbial mat preservation, which creates these textures, these layers, sometimes towers up to 6 or 7 meters in height. Stromatolites. Stromatolites are the dominant fossil evidence for life on Earth for 3.8 billion years. I mean, they're just the dominant fossil evidence. And here they're living today still in places where there's no grazers, there's no fishes to eat them. So this is at Shark Bay in Australia. This is today's stromatolites. And then here we are at 2.7 billion and 3.5 billion, climbing back. So here's the model, graphically easier to understand. So in the ocean environment, if you have material falling in, it just dilutes in this massive pulp, which is full of divalent cations, it's full of salt, and this is extreme, right? An absolute extreme environment. So all this contribution from space, meteoritic contribution, volcanic gas or particulate contribution cannot be used in life in order for an origin of life in the oceans, but it can if it falls and concentrates in pools. And these are clement conditions. So the optimal chemistry, or the Goldilocks chemistry that we have in the lab, and all of our colleagues do in the lab, is something like this. It's a little warm pool. This is in Kamchatka in Russia. It's a kind of warm little pool between 60 and 90 C, and it fluctuates, and it has material concentrating and falling into it. And this is Sutherland and Powner's definition. And so life can get started in this clement environment, this optimal environment, cycling. We'll see why cycling matters. And then it adapts to extreme environments, dilute rivers and lakes. The riverine, lacustrine, shallow marine is extreme because of salt and tides. And so you have this gradient, this massive gradient of collecting starting materials, concentrating them, cycling them, adapting them to stresses, and then distribution all over the Earth. It's a logical downhill flow for early life, but an uphill adaptation gradient. So this is how it started in the 1980s. So Dave Deamer, my colleague at UCSC, is one of the world's greatest living membrane biophysicists. And he had invented, there's a technology in the market called Nanopore Sequencing from Oxford Nanopore. It's called the MinION, and that's Dave's invention. We'll talk about that a little at the end of the talk. But this is our laboratory simulation chamber for doing wet-dry cycling. So we can dehydrate a solution as it rotates around in a vial and rehydrate it. We're doing this following reaction. So in here, we're putting the building blocks of, say, of RNA. And if you notice, when you form an ester bond, you actually have to have water leave. That's why it's called a condensation reaction. It's the only chemical diagram we'll show so I don't cut my readership in half. But if you sandwich, thank you. If you sandwich, these clickers always have a convenient button to stop playing. So if you sandwich stuff in between something called lipid or amphiphiles, you create a two-dimensional phase. So when you're dehydrating a solution, you go to a bathtub ring phase, and there's filmy, soapy stuff on the bottom. That's a chemical factory. That can do this reaction. That can stitch all these polymers together. It's fantastic because water's drying out, it's leading through the bathtub rings into the environment, and suddenly you get this massive production of random polymers, like a giant roulette wheel. So then when we look at it and scan it under, or analyze it, we get all this product. We get all this RNA, long, long chain, and this is through cycles, 1 hour, or 4 hour cycles, 1, 3, 5, 7. We can get more and more product, and this is when we put it through our nanopore, because David invented this 20 years ago, but now it's a product, so we can use it for origin of life research. We can just dump our RNA into the nanopore minion, and then we see these blockades as it goes through this pore, and we see the signature of RNA. And recently we've done DNA this way. That's about to be submitted into publication. It's a huge result. It's the self-assembly of DNA in the natural realm. Quick question?
Yeah, in the pond, where do the lipids come from?
Space. Everything is coming. Yeah, I'll show you. Everything is coming from asteroidal material, the feedstock, and some of it from the hydrothermal system. But in this period of time, you had between 100 million and many billions of times the amount of dust in the solar system coming into the Earth in abundance, and basically concentrating in the ponds. So vast amounts. So you don't have to fix the carbon. You don't have to make amino acids if they're falling from the sky. So it's a complicated question.
Yeah, quick follow-up. What other chemical conditions need to be present for this spontaneous creation and assembly of RNA?
You need fresh water. You need the concentration of the material where it can leach out. So what we do is we take...
I'm sorry, by the material you mean?
the starting, the fatty acids, the nucleobases, and the amino acids, and various and sundry other things, metals and things like this. And we actually take pieces of this is the Murchison Meteorite, which is older than Earth. It's 4.6 billion years old. So we take interior sections of that, grind it up, and sprinkle it onto our surfaces, and then we cycle it and get the polymers. So we can do it we get membranes and polymers. So we can do it with this material that's falling in your gutters, every day still.
What protocol do you use to prevent contamination?
Sorry?
What protocol do you use to prevent contamination?
We're doing synthetic RNA, so with only 2 building blocks. So we can always tell real RNA from synthetic. And also in the hot spring environments, we don't generally have any biological material intact. Like the one I showed you at Hell's Gate, there's nothing in there. It's the self-sterilizing, which is handy. Those are good questions.
Wait, if it's self-sterilizing, then we can't, what's happened? Life's moved on to greener pastures?
Well, it turns out that the chemistry, the regime for prebiotic chemistry is still in the hot, extreme environment. It's still in the extremophile environment to get the polymers to form. It's something like 70 to 90 Celsius. And in this case, it's slightly acidic. And you will find life forms in those environments. But then I think it adapted to lower temperatures, to capturing sunlight. You'll see a little bit more of that in a second. So this is trying to do it in a fumarole vent at Mount Lassen at Bumpus Hell. And that's me on camera holding an experiment in a vent gases. And the vent gases hydrated and dehydrated as they wafted along. So this is one of our first field experiments. So we got a little bit of RNA. But this is an analog, but it's not the analog we really wanted. So that's why I started going to these field sites. So this is Yellowstone National Park. That's a siliceous hot spring on Buffalo Meadows. And what's happening here, and this goes back to your silica question, this is silica coming out of solution. And it's this gelatinous kind of thing. It's mineral precipitating out of solution, becoming hard sinter. And it turns out that all life in the hot spring environment like this it's about 90% silica by dry weight. So microbial communities form strands and balls and sheets and everything, and they use silica as their building block, as their structural element, to protect them from UV and to hold them together. And so it's it was probably silica coming out of solution was probably vital in the early phase of life on Earth. And it turns out that if you look downstream, so you start here, it's abiotic. This is erupting about every 45 minutes. And you can see the layers forming here through the wet-dry cycling of the silica. And then right about here, you get orange photosynthetic bacteria, and then you get the green further down. So there's a temperature gradient from this particular pool. So this is all chemical feeders here because it's too hot for photosynthesis, and this is more photosynthetic further down. Because it's more diluted, you have to make your own food if you run out of food. Not enough chemicals to eat. So that's actually the solution from a year ago. And we were testing that we could form vesicles, membranous vesicles in these waters. Because the geologists challenged us and said, you'll never form vesicles in silica or acidic conditions in, say, somewhere like Yellowstone, because such and such won't work and such won't work. So I went out there and we did it. Sent him the paper, actually. So here's that result. So this is vesicles formed in Yellowstone waters and encapsulating DNA through wet-dry cycling of those same waters. And this is what seawater does to them, just create crystals. So if you try to wash your hands with soap in seawater, it turns to curds, right? So you can't form the building blocks of the protocells, of membranous vesicles you need to have to put around your polymers to get the system of selection going in the sea. You just can't do it. And it turns out that the composition of the cytosol in all living cells is much closer to Yellowstone's composition than it is to seawater. And basically life had to evolve massive pump systems to get salt water out of cells. So if you, question?
How come the pH range is so wide?
Oh, in environments like
1.3
this?
to 8. Or like, here you're mentioning that it's an acidic environment, but it's all the way from 3.3.
It's hydrothermal systems are just like that. You can have even interconnected pools with a pH will be 9 over here and 2 over here, or 1 over here. You can get pHs that are as far down as 0 in some of them. It's all water-rock interaction that does that. It's an amazing chemical factory in a way. So this is what happened last month in New Zealand. You saw the video, and that's how we did the solutions. We put them in vials in a heat block inside the 90 Celsius sort of spitting pool, and we used acidic and alkaline waters to cycle. So every hour I was checking the vials to see if they were dry, and they look like this. So there's our film right there, and checking it, see if it's dried down, squirting 0.1 mil of solution back in, and then waiting, and then drying it down. So I did that 4 times in the environment. And you saw on my phone that the amount of polymer we got was just tremendous. This is not in direct contact with the sediments, and that's our next challenge, probably in February, is we've identified sites that are fluvial sediments, and show you even, where we can make these protocells in situ, directly in contact with it, without having stuff being washed away all the time. So fluvial environment probably be the first. So then what we do, because we can dry down and make those films, make lots of polymers, when you add a drop back in, so those vials that I was doing, I was making at least 1 trillion compartments in that one little vial, some of which have random RNA in them. And we stain them, this is what they look like. So this is just sort of the composition. So what that means is you can do combinatorial selection and boot up life. So as Eric was actually saying, at the end of his talk during the Q&A, he's made the following statement that somehow we need a selection system that became before life that could then act on a unit that then could bring the functions of life together. And this is what this system is. So it's pretty fresh and new. So for all the nerds in here who's been assimilated, you were all assimilated a long time ago. In 1981, when I was doing computational stuff, I was like the weird kid in my town, right? Like the computer person was the weird person, but now we're all weird. So, so the metaphor for computer science, why you don't need a creator to bring life into existence, is you effectively, you could write programs in the following way. You could have a random paper tape puncher.
Does anybody here besides you and I know what a paper tape puncher is?
He knows what paper tape is. You've all heard about punch cards. Paper tape's more primitive than punch cards. Way more primitive. The homebrew people that had to work with it. So actually, I have a big collection of vintage computers, and this is one of them. It's called an Altair 8800 from 1975, and its initial interface for programming, apart from the switches on the front, was paper tape in a device called ASR33. And so, say for instance, your origin of life metaphor is an Altair. It's got an energy source and it cycles, and you just feed it random tapes, random short tapes, and it runs them through this thing called a CPU that just runs them, and frankly, most of them crash. They crash the system, and there's no OS or anything. It's like most of them do nothing, right? There's random programs, but some of them might do something that's selected for, like show the blinky lights going or something. And so then that's selected for by, a form of nerd selector. And then what you do is program A is then conserved, and then the paper tape puncher just generates B, C, and D, and you attach them and run those. And slowly, very, very slowly, we'll generate perhaps a new computer that has more functions if it's actually influencing the hardware, a screen and keyboard, and maybe A, C, and F. These programs stacking up might make you a laptop, and this one might make you a smartphone. So it's the evolution of software and hardware together. Let's see where we can find this in nature, the same system. So it's a very inefficient way of creating code. We pay engineers good money to do it slightly more efficient. Slightly more efficient. But sometimes they're just doing random paper tapes. and charging billable hours. So Y2K, need I say more? Okay, so where we find this in nature, this environment, is the random paper tape puncher is that polymerizer. It's that bathtub ring thing that's making gazillions of random polymers coming from the organics from space. Those polymers are then fed into a primitive life computer called a warm little cycling pool. Here's our Charlie Darwin. It's got a source of energy. It's got a cycling thing to test each of those packages, which are
The final result, PowerPoint.
Final result, the group think of PowerPoint. Okay, so here we go. Protocells, yes, these are the programs that life tests, random programs. It's membrane encapsulated polymers, which is what you are basically, still. So our Charlie Darwin is the nerd selector, and he decides which survives and which doesn't. And the initial criteria is, did you pop or not? Did the membranous compartment fall apart when the pool was filled up again? So it's really sort of a simple thing. And it's the evolution of software and hardware together. So let's take a look at it. So here it is. Here's our three-way cycling, dry bathtub ring films. We have, when we put a drop in the pool, it forms these protocells. They bud off. I'll show you what that looks like in the microscope. That's what the dry films look like. That's what the budding off process looks like. Stain to show the encapsulation of the polymers. You test them for stability. They're just now being subject to shear forces, and these flaky, leaky membranes tend to fall apart really easily. The stuff that comes from space is actually very fragile. So guess what? A selection thing happens, and the polymers that are just right, like this one, to help stabilize that membrane, those polymers come back down and they form a sludge in the bottom of the pool, and they're your survivors. And the sludge, this is the real leap. This is the real insight. This sludge of surviving polymers is actually, we believe, the progenote, the Carl Lowe's, George Fox progenote. It's the unit at the base of the roots of the tree of life. And this took a couple of years. I did thought experiments to solve this one. It's like I said one day to Dave, wait a minute, there's a moist phase in between a wet and a dry phase. Like, duh. And we were driving in the Australian desert, and it just had a cyclone to come through. And the desert was just rife with life. It hadn't had a rain in a year. I said the moist phase is where the metabolism can kick in, just like what we see after a rainfall and after a dry period. And that it exists in our model because it's a collection of protocells that manage to survive one cycle. And guess what happens in a moist soil or a moist phase? All kinds of products get moved around and shared. There's all kinds of communication. We're here because we're grazers on the microbial map that is the moist phase. You know, that dominates life on Earth. Is this moist phase progenome. But the progenome is the unit, it's a computational unit that could, through a huge amount of cycling, boot up every function of the living world.
Bruce?
Question?
Yeah. Does that also mean that you have to select for survival during the dry phase when all these membranes presumably get messed up?
Priyan asked a very good question. Does that mean you have to select for survival during the dry phase? Yes. In fact, at each one of these phases, there's selection criteria happening. In the dry phase, it's competition for monomers. So it's like a Conway's game of life kind of a thing going on that makes polymerization emerge. This one is stability. This is basically selecting for stabilizing properties, and this one is selecting for metabolic properties here, just before the whole system dries back down. And so you get this very sophisticated selection engine in a little dish. Question here?
You talked about before cycling the thermal regime, right? So now we're talking about cycling the wet and dry regime. Are these orthogonal or are they supposed to be coupled?
They're coupled. So the question is, what do we mean by cycles? Well, it's cycles within cycles. So as the sun rises, you get diurnal cycling. You get wet, dry through just the sun evaporating. But you get water pulses in hydrothermal systems. And it turns out that if you make a bunch of polymer in the lab, and you put it into an aqueous solution within, say, RNA, within days to hours, it is toast. It is hydrolyzed, it's depurinated, everything, right? So life has to continually remake polymers, constantly, and correct for errors. And so that's what life still does. So this system does that by simply resynthesizing them, maybe through templates. So if you're in a deep aqueous environment, you can never do this. You can never form the polymers in the first place because you have to get water out of the system. And then if you've made like a dimer or trimer or something, it would just get hydrolyzed right away. There's just no way to do it. So that's why the ocean origins are no longer in favor in the origin of life community as the majority of them now. Question?
But you don't yet have autocatalysis in this stage?
Yeah, so here's where we can all use your help. Because this is a huge and wonderful intellectual challenge. And I'll just go on. So this unit, which can be made in the lab, and you can use synthetic biology techniques to trick it out. We predict, so I did put the slide in for this, but Dave and I predict we're going to get functional polymers drawn out of the selection system. First one is stabilization. The thing that stabilizes its vessel goes back to live another day. The next one we predict will be a pore-forming polymer. So just a random polymer that jams into the pore, which allows stuff in and out more easily. So it does stabilization and it prepares you for metabolism. So if I've got a progenome down here, or a mass of protocells that has a bunch of pore attached or membrane attached things, as the pool level drops down, this is all physically based, you get more and more concentration as the meniscus drops, and stuff will start forcing into the protocells. And this is where I'm talking to Stuart Kaufman and all the people. We think that metabolic cycles could kick in this phase here. It's all the right conditions. Now those metabolic cycles have to be selected to make more stable protocells in the future, but where they would really come into place is template-based replication of polymers. Might be one of the first ones. So it's a huge thought experiment space. I mean, I think about this all the time, but I don't have the background really to visualize this. Because after you get pore forming, you get M polymers for metabolism. Then you'll get C polymers for catalyzing metabolism. Then you'll get R polymers for replicative metabolism. Then you'll get D polymers for duplicative metabolism, which will lead finally to the division of the first protocell, which is the transition to life. So let's take, this is how it all comes together. This one took 2.5 years to build. So we came up with this hot spring origin of life, synthesis of the most of the building blocks in the solar system, delivery, exogenous delivery onto land, onto many, many pools of many, many pHs and many sizes and cycles. And that's a huge combinatorial selection system. So the concentration of that material in these pools start generating more complex organics like membranous structures. And then one day, a pool is fed on a continuous basis through the system by, this is how we do it in the laboratory, we don't have the budget, right? Is a cycling pool that's driven by geyser action. So you get the temperature pulse, which helps your chemistry. And if you actually have, double helical things, it's gonna unwind them. So it's a natural PCR. And then you have this system that's in continuous cycle. And it's an evolutionary system before we have even genetics. So you're just dealing with background phenotypic emergence. I'm having this long conversation with Freeman Dyson about, he talks about this, looked at this model for about 10 years, and he talks about we could get a long way on phenotype before we need genotype. And we're saying we need genotype early. Question here.
They just assembled a model like years ago. There was a tide pool based, right?
Was it what?
There was a tide pool based one, sort of at the ocean's edge, and the idea is you would have this top, this rock where water would collect, and you'd have everything. Basically, the tide would come up, refill it, and you get the more and more concentrated.
That's also mentioned, but you have massive salt concentration, so no membranous compartments, no polymerization chemistry either in those environments. So that's kind of out. It really matters what's in the water. Turns out. So then, here's the money shot. Is where here's our call it an origin pool. Just like in Yellowstone National Park, these are chemical feeding, right? They're being fed by exogenous sources. As soon as this material, which has the property of distribution, you can wash it to sludge, right? You can wash it downstream real easily. You can see this in Yellowstone today and in Australia, where you have these storm events that take microbial mat and throw it into other pools or blow it by wind like hundreds of kilometers. So microbial mat distributes aerially on the Earth today on a massive scale, in the high atmosphere, some of it. And this one, having distributed downstream, it hits a dilute lakeshore or stream. This is the point it has to be making its own food. So we predict that photosynthesis comes in somewhere out on the outport of a system like this. It has to be, because you have to be an autotroph, a photoautotroph. And so we're predicting that the actual transition to the living world happens here. The transition to cellularity, where instead of these disappearing and reappearing, getting replication for free, one day, two of these things learn, or one of the things learns how to divide itself and create daughter cells. And that's a whole other amazing process. Like how, so we have a combinatorial system called a progenome that can lift the functions of life in a community in a network effect. And it goes up toward the most important moment, which is cell division. Right? So cell division is the really huge, titanic problem to solve in life. And for about 30 years, I worked on this, working backwards, getting the most minimal protocell and saying, if anything that's on the path to this thing dividing is part of the story of the origin of life, anything that isn't is not. That's an Occam's razor approach. So we think that life starts somewhere there, and think about it, the top layer, say the top layer of cells, if they learn how to capture sunlight's energy, right, they learn how to do that, they're energized, right? They've got tremendous metabolic potential in them. They're probably the first dividers. The origin of life probably happened to the epithelial layer, it became cyanobacteria, right? And they may have started with simple pigments like polycyclic compounds, black pigment, which were also falling in from space. So you got your sun capturing pigment for free even in those environments. So once you get this transition to microbial community, they have to adapt to the saline estuary environment, tides, massive amounts of change in pH and salinity, and they get more robust, and then you get marine stromatolites. So it looks like this, and then the rock records support it. Here's a hot spring life, here's lacustrine, riverine, and here's a yeah, somebody can debug this. I'll just go to the next slide. And the marine stromatolites tend to be big domical things, as you saw. Yeah, just go to our marine stromatolite. They're big domical things. The next slide. So this was the place. So we've basically been working on this for 3 years, and Scientific American approached Martin and with the find in the Pilbara of the hot springs, and developed this cover story. We didn't think it would be the cover story. We knocked the eclipse off the cover last year, and so one of the team wrote and said, well, "Why did this happen?" And the editor's a microbiologist. So she actually said, "We think this is one of the most important stories we'll publish in this century." Wow, pretty cool. Okay, next slide. There's the cycling system. So this is what Scientific American did with it. So you saw my treatment. They did this really cool thing of, here's our start point, and here's the drying, and here's all the layers that organize the monomers. Here they are budding off into protocells. Here's the sludge that returns. More stuff is made, more sophisticated protocells, until you get to a unit that's capable of evolution on its own, the progenome. And then that climbs towards cellular life, and on a massive scale. So that's the sort of the summary of the work we've done. Experimentally, we proved all these things, that we've come all the way down to this point. The conjecture is the progenome itself. So we're forming an institute now called the Biota Institute, trying to find about $100 million to get it going, and it will generate a lot of seed technology and company. We'll tell you about that later. But one of the things it will do is the Progenome Prize. For teams that are cycling these protocellular aggregates to see what polymeric evolution can draw forth. So the first team to show a pore, a functional pore, wins the Progenome Prize of some unspecified prize. So that's one of the things we're doing. So this is conjectural, but we need computational people, we need A-life people, we need combinatorics people to model this stuff, because it can now be tied to something you can test chemically. You could create through synthetic biology basically a little protein that's short, throw it into one of these cycling systems and see what it does. Does it have a role? You could do it with synth bio, not waiting for de novo random sequencing. So next, thank you for doing the slide. So these are our next candidates. This is for February. Hopefully I can get the budget to go back. This is Hell's Gate. So next slide. This is a lagoon environment. So New Zealand kind of looks like the prebiotic Earth. It's an amazing thing, with lots of sheep. But this is lagoon environment, wet, dry cycling all along here. This is all stromatolite. This is a little pool. It's pulsing with waves constantly. So it's another cycling system. We go forward. This is the candidate that I've put my money on, my poker chips on, for the origin scenario. Braided river systems that are infused with hot springs that keep going. So that's one of them. This is what they look like. So where there's landscape without a lot of tree roots or plant roots, you get massive erosion. You get these massive braided river systems. And in this period, there would have been hot springs poking up. This would have been steam vents everywhere. That is the perfect environment as a bioreactor. It's massive in scale, trillions of individual environments. We keep going, so there's another one. And we're going to use one called Parariki Stream. I think it's the next picture. There it is. So that's the environment. That's the place I'm going to put the reagents directly into the environment. And here's stromatolites forming in Parariki Stream in New Zealand. These are called fairy castles.
What's the scale on that?
That's probably about an inch. Those are about an inch high. I think I was holding one before in the video. So keep going. So this has been incredibly well mapped. And these hot spots, which are all over here, don't change for decades. It's a point bar. So streams are going past It provides a washing function to elute waste. It provides a perfect chemical environment for thousands of gradients and compartments in wet-dry cycling and different environments to try the chemistry. So it may be that in 2019 that we make copious amounts of protocells here, right, directly here, we can make them. And down here we can't. So we can test null hypotheses all over in one point bar. We can test the whole thing. It'll be the Miller-Urey experiment of the 21st century, really. So let's go on. And this is all, that's all too much detail, so let's go on. Okay, so, because there is another talk, I don't want to completely melt your brains. So we're forming this institute because we now have a testable hypothesis that's showing great promise, but also spinning off startups. So keep going. So we call it biota.org. So one of the first spin-offs from Origin's work, which is the most financially valuable, is basically Oxford Nanopore. So here's the device that resulted from Dave's work in the 80s, where he literally was able to pull RNA and DNA through a pore in an artificial membrane, and it would count through blockade the voltage changes. It would literally count the bases and tell you what the bases were. It's a fantastic technology. It's like, look how small it is, right? So Illuma's been suing. Right? That's what happens. Is this is disruptive for Illuma's business. So this is a prior example, probably valued more than $2 billion now. The next startup that we're doing. This is the patent. That's the previous patent. It's based on. We filed 2 more patents. This is called UpRNA. And go on to the next one. And so this uses our wet-dry cycling system. Because it turns out that, next slide, that if you can produce short pieces of RNA that sort of get into a cell, guess what it can do? It can do a silencing action. It can go to where viral RNA is working in the ribosome, getting the cell to trick it to make more copies of the virus and shut it down. And it's totally targeted, it's totally directed. But it's very expensive to make, say, a 20 mer of RNA with enzymes. It's very costly, you get extremely low yields. And one day Dave woke up in like December and said, oh my God, we're making them in their quadrillions. If we can figure out how to make it with a template, and maybe next slide. This is what actually happens to these short silencing RNAs. They sometimes get grabbed by a great big protein called an argonaut, which then floats around and then it connects in with the viral RNA and just turns it off or breaks it up. It's amazing. It's a universal shotgun method to stop all viral infections. And this is the device that we cranked out. It looks like something from the Victorian era. But basically, Dave said, well, we did the wet-dry cycling little dishes. What if we had a cylinder? And we worked up this over lunch one day. The cylinder would go into the bath, pull up the lipids, pull up the monomers. It could be any monomers you want. Come up to a heat gun at the top, dry the whole thing down, and then go dump them back in. And so he built this on campus, and it works. It generates vast, continuous quantities of short oligos, they're called. And with what we're doing now is putting in a template so we get a predictable one every time. And then in May, we met with an exosome company here, which several of you were at this talk, and a company that makes exosomes, the little blebbing off lipid compartments that cells continuously produce. And they could be for waste removal, they could be for communication or putting products to other cells. And what we would do is, if we can perfect this, we can put a continuous stream of short interfering RNA through microfluidics attached to the exosome stream, wet-dry cycle to put those inside the exosome, and then dump it into a viral infected cell mass and watch if it shuts down the virus. And if this works, it'll be one of the great medical innovations of the last 500 years, because we can target all virally influenced disease and specifically turn it off. We can use conserved parts of the viral genetics. So, there's no way for evolution to respond. So we can cure the common cold.
Yeah.
Yeah. Question?
Can you briefly describe how the templates work?
That's all under patent. That's all, we can't talk
Or
about.
just how templating mechanisms for...
Yeah, that's Dave, and we don't talk about it.
Even in
You'll have to join him.
even in the hot springs when the...
We're not doing the templating yet in those environments. So this is like a startup company where we're doing synth bio techniques rather than doing de novo synthesis, which is hard. You have to wait a long time. Question here?
Yeah, this goes back to much earlier in your talk, but I wanted to wait to see if you would explain it. So I'm not clear on what role the RNA inside the cellular membranes plays in the very, very early life, where basically all you have is the RNA and cellular membranes. Like, is that encoding to produce something else? Is it acting as
could
an
be.
enzyme?
Yeah, that's a really good question. Sorry for not covering that. So
Yeah, no worries.
Thomas Cech in 1982 created the first ribozyme, which was a short RNA that both had a templating function to make a copy of itself and could fold and do a function. So then it started the RNA world approach, which was we just didn't have to have DNA. We had RNA was the sole functional polymer, and then it would couple into peptides. Because peptides are being co-synthesized in the environment. So the interaction of RNA and peptides, which gives you the ribosome, gives you a lot of goodies that they thought, okay, if we just get a prebiotically plausible RNA source, a selection machine, and then peptides interact, which are also being selected, we could boot up a chemical circuit that would look like biology.
Cool. And then in terms of ribosomes, it's been a while since I last took bio class, but I remember them being incredibly complicated and essentially being able to go straight from an RNA sequence to an amino acid sequence that's a protein. So when you say ribosome, it sounds like you might mean something different.
The ribozyme is like a primitive version of that in one molecule.
All right.
So the people who are studying ribosome fossil, so what they did is they took this the ribosome is this gigantic multi-billion atom machine that runs every one of you right now. It runs all life pretty much. And it just does that transcription. It takes the data store and pumps out proteins to make us all do things and live. But it's a gigantic system. And so what they're doing is, especially at Georgia Tech, they're taking apart the ribosome. They're actually predicting when the parts evolve. And the earliest pieces is this clamp, which is around this PTC tunnel where the stuff is going through. And they think that's the oldest one. That's probably 4 billion years old. It's a fossil. And what the clamp did, so here, picture flimsy, flabby early life that barely works, right? Doesn't have a ribosome, doesn't have machinery like that. Yeah, we built that. Wow, that's pretty cool. So you don't have this efficient processes. Everything is like a Rube Goldberg machine, or even worse. I mean, Rube Goldberg machines are sort of predictive. And you have this rickety early chemistry. That's why when the hydrothermal vent people talk about the need for massive proton gradients and whatnot, they're talking about modern life, not prebiotic systems or early biotic. You don't need any of that. In fact, that's very advanced technology to do these gradients. So you have this primitive system. So the thing that became the ribosome was this clamp floating around, and its only job was to take the peptide and keep the ends from attaching to each other. It's called circularization. That's the only job it had, and it had this job for 100 million years, but then stuff kept getting added to it. And then it built up this bigger machine, was able to do things really well. And somewhere around probably 3.9 billion years ago, ribosomes were Brownian machines. So the ticker tape of the RNA is going through, but it goes backwards sometimes, and it generates crap. More than 50% of the time. They're Brownian machines. And a late cofactor was a ratchet that would pull that RNA through predictably. That was a late cofactor. That's a really late molecular cofactor. So our ribosomes worked for shit until that thing was added. And then life got a huge boost because this cofactor was like, hey, you tell us what you want, we'll make it. Predictably. But that was not the case. So there's going to be an entire new biology emerge of rickety early, what might be called progenote biology or early life biology, that are really realistic models of the pathway from there to here. next slide is, so this is my concept coming from this unit to the next one, which is what we can do now, because we can make all these feedstocks, we can make a chemical search engine. We could make a system with microfluidics or nanodrop chemistry even with an AFM tip. It's a whole other discussion. We could make systems that can find catalytic chemical circuits through massive search. And this is a project called the Genesis Engine that will instantiate inside the Biota Institute and look for partners. But my neighbor in New Jersey, I have a house in New Jersey, he's the head of painkiller research at Glaxo, and I showed him this origin of life model and cycling systems, and that we can select for things and run them through nanopore sequencing in real time, and then flush the experiments out again and restart them, so we can do combinatorial chemistry in some future technology. And he said, I don't give a damn about your origin of life thing. That's the hardest problem in chemistry and biology. What I care about is this search capability for pharma. I said, what do you mean? He said, because we could dial in this, we want this pharmaceutical, and it would find the shortest path and the highest yield way to make it through these catalytic cycles. So I said, what's that worth to Glaxo? And he said, we would invest $1 billion to get that capability. So if they do that, we'll get throw-off, hand-me-down machines that we can use for unfunded origin of life effort. So next slide. So that's the idea of a reactor and a big soup. And so you can tease stuff out of this basic science that's totally fundamental in doing chemistry in different ways, in marrying chemistry with computation. And go to the next slide. Do I have anything more? There was another slide I was going to show you, which is, this has just come up, and if you go to the SAND conference or Science of Consciousness, I've been actually doing talks at these conferences. And strangely enough, so Stuart Hameroff invited me to Science of Consciousness last year, and I thought, I had never cracked a book open on consciousness. And I then did crack a book. It was just all mumbo jumbo. Right? It's like, oh my God, it's all words, right? I can't get into this. I cannot mainline this field and do a talk. But what I did do was I started doing more thought experiments. I'm like, how does the origin of life relate to consciousness? How do they relate? How do they connect? So that I can not sound like a blathering idiot in front of Deepak, although or others. So it has to have woo but goo. So then I did the following thought experiment. Like, what is that protocell system actually doing? If you were to pull its basic properties out, what does it do? And I'll do it with hand waving. Here I'm criticizing our colleagues for hand waving, but here's a hand waving. So here's the background physics of the universe. You push down here, stuff rolls over here. It's pre-statable. You know, this is Stuart Kaufman's language. Maybe not at the quantum level, but I mean, at the classical level, it's pre-statable. Well, as soon as you get a little compartment that opens up in that field, you change probability, right? Stuff is crowded in there, like our little protocells. It increases the probability of improbable things, like chemical reactions, right? It vastly increases it. As soon as you have a compartment that's permeable, property number one. Property number two is when you get more of those compartments that have these improbable reactions happening in them, you get a network effect. Because a reaction over here diffuses a product to go over here, which triggers something which comes back. The network effect, we know from the internet, and from our whole society, our whole civilization, is so powerful because it can lift complex things into being that an individual serial action cannot do. And then you get on top of that, so you have probability, interconnection, or intermessaging, and then a third thing can emerge, which is a memory system. So what we're doing right here is we've crowded ourselves together in this room. I'm doing a lot of hand waving and I'm doing a lot of interconnecting. Conversations are happening that are improbable in any other way. And a memory system is recording this to go out on the net or whatever for the next evolution of the idea. So I put it to you that this probability begetting interconnection or internetworking begetting memory, which makes improbable things even more likely, which then makes more interconnections, which is the engine of creation. It is the system that describes how everything is made, from biology to economics, to the way politics works, to the way technology evolves, and maybe even like spiritual experience, which I was trying to get across to Rupert Spira and JP Sears. And JP knows everything already. He's Altair spiritual, so it's impossible to put anything over on his guy. But so this PIM thing, we're now working on with people like Jeremy Sherman and Terence Deacon and all these other philosophers. It turns out philosophers are interested in the origin of life. I had no idea they were. Daniel Dennett writes about it. But they don't work with chemical models. They don't work with anything that's actually in the world, right? They make up stuff. So we're now actually working with them. We're now starting to work with the groups in AI. Because it turns out, and I did 4 conferences on this theme with AI and artificial life people in the 90s. And AI is like stalled, and so is A-life as a field, because we're using procedural algorithms that break, right? Even really the only reason we have something we call AI and self-driving cars is that microprocessors got fast enough. Because AI is nothing more than really table lookups that are done really fast, right? There's nothing, there's sort of learning through neural networks, but it's just more table population lookup. There's nothing to it, right? I left that field because I thought, oh, it's a dead end. Code and other laws of cyberspace is like, this is not interesting. And I started studying this because you have a completely different environment. You have a massive bunch of bouncing ping pong balls at the low level, pushing shit around. There's no procedural things going on. It's like all probabilistic. It's a fantastically rich physics in which to immerse things. And then you have this probability engine, interconnection, and memories being written constantly. We could probably develop AI on these principles that uses evolution at its core for becoming, and writes its own code, just like the computer science example. So if you know any AI people that want a new model, and that means a new model for silicon, it means a new model for computing and how to think. But computing people hate no ops, right? No operation. But nature relies on no ops as its fundamental physics. Like nothing happened, nothing happened, nothing happened. Something happened, nothing happened. And it creates a rich enough tapestry for life to power up us. So that's, I know there's some nerds in the audience.
Sorry, I'm not clear on that. No, I'm serious. I do AI, and I'm not, your metaphor isn't quite working for me, so I'd love to understand it.
We should talk, because this is just a new idea that we could use this model somehow to inform AI. Yeah.
Okay, yeah.
I would love to talk, because I don't know, I haven't really cracked a book on AI either. But it seems to me that if we discover the system that can power out of the background of physics and make the system away from equilibrium, that's self-adapting and evolving, it'll teach us about everything else. But those fundamental 3 principles, I think, can be used in anything. So I think we're done with phase 1. The talk phase 2, the 2 goals I took on was about 14 or 15. I thought, you know, I'm a nerdy kid in a non-nerdy town in Canada. I'm really interested in the origin of life. I was doing thought experiments in those years. But I was very interested, there was the peace movement and the ecological movement was going, right? And everyone was marching in the streets and things. And I looked around, I'm a very pragmatic little kid, like, nothing's gonna slow us down. We are gonna pave over that landscape, and we're gonna build suburbs here, even in our little town, we're just gonna pave the whole planet. Nothing's gonna stop us. No good idea is gonna stop this thing and slow it down. So what we need to do is to extend the biosphere, to allow the biosphere a place to reproduce, which is the near solar system. And so I was working since 1978 on ways to harvest asteroids. And you'll see the interesting, sort of surprising solution that the collaborators came up with in the next talk after the break. But are there any dying to ask questions before we go where we boldly should not go?
I have a question.
Question over here.
Yeah. There's an anecdote that you made in the video from New Zealand about how it wasn't a supercell that was individually responsible for the genesis of life, but rather collaborative groups. Can you weave that into the end of what your conclusion here? Because I'm not sure I'm seeing how that.
Yeah. So I was literally on the bus with Dave. And some of us did get on the bus when the bus came by for different reasons. I said, Dave, wait a minute. The argument against, the creationists use for irreducible complexity, right? That in the Hadean, this is 4.2, 4.3 billion years ago, you have these flabby fatty acid membrane enclosed polymeric systems which might have the ability to sort of work against their degradation slightly. But if you have one of them in the pool on its own, it has a dwell time that's very short. So that's why I proposed that they all came into an aggregate, which formed a niche. The first ecological niche was the aggregate of clustered together robust protocells that could protect themselves against pH changes, shear forces, temperature, everything. And that this unit would be more likely to survive. The polymers within would be likely to survive because they're protected. They're like clumped together for mutual protection. And Dave said that'll be selected for. Individuals can't do it. They're just too flimsy. They don't have the technology to survive outside of the community architecture.
So it's like a protocell group of penguins on a ice
platform. Yeah, like one lone penguin trying to get to the South Pole. And we are, so in effect, the entire story of life from the Erdot is still all about communal action and sharing. And they've now discovered that forests, even between species of tree, so in the pinch of forest, in the redwood forest here, there's 7 miles of mycelial tubing, 7 miles. And that tubing is connecting all trees to all other trees, irregardless of species. And those mycelia tubings are supplying all the minerals that the trees need, and the minerals are supplying all the sugar that the mycelia need. And the mycelia are transport mechanisms that actually heal sick trees that have a need, and they'll transport material for kilometers over mountain ranges. It's amazing. So we've discovered that, the idea of trees competing with each other, trees are in this giant network of sharing that increases the health of the overall forest constantly. So in a sense, it's still a progenitor planet. And in another sense, if the survival of the fittest was a meme suggested by Herbert Spencer, that Darwin put in his book, the 3rd edition, I think, of Origin of Species, and didn't like the term, right? Survival of the fittest, very Victorian term, to replace natural selection. Because Spencer was telling Darwin, well, that implies a selector. Use this handy Victorian term, survival of the fittest, from my writing, and that's created a lot of grief. I mean, that it certainly went out of science a long time ago, the idea, but social Darwinism, bad behavior in business, bad behavior in politics, it's a story of separation. So in a sense, hardcore reductionist goo science, not woo, but goo, can show that the only plausible way by which we came into being was a collaborative network system. That's the fundamental unit. And we are that unit. And the health of that collaborative network system is the way we survive or don't. So we can then start to break down those that would like to create separation, fear, and anxiety between groups to harvest for their own gain, which is part of our species, doing it all the time. So all the fake news and all the craziness, right? So that's where, I'm not a philosopher, I don't play one on TV, but this idea coming out of science is maybe as powerful as relativity theory was in the 1920s that helped modernism boot up, Einstein's relativity theory from the eclipse of 1919. Perhaps the idea of the symbiotic start coming, for instance, in 2026, if we get funding, we'll have 4 or 5 teams making progenotes and cycling them. And if there's a time-lapse camera on those dishes, here's the Turing test for the progenome. We're not talking about life, we're talking about the unit that can get us to life. The sludge is growing with each cycle because polymers are emerging that stabilize the bubbles. And then maybe a pore forming one is followed by a little metabolic one. Still no genes yet. but you get a sludge growth, and then with not any change, you get a crash in the population. Because maybe they've run out of product. They've used products or the building blocks up too efficiently. And then the sludge grows back. So it adapted to some of the stress. And you just pipette that and sequence that, nanopore sequence that, and see what polymers emerged in that system. Like, that'll be cool. It's like this system adapted. We can do this with bare naked molecules now. We can do polymeric chemical evolution, it's called it. It's been done many times. Can we do it within protocell compartments? That'll be very, very suggestive of a system that can lead to the living world. We'll never know exactly, but yeah. Question back here.
Yes, I had a question about ribozymes. Ribozymes originally, I think, were conceived to actually do metabolic, like enzyme type things themselves, other than just sort of create ribosomes or primitive ribosomes that then make protein enzymes. And I was wondering if, in your conception, these ribozymes are actually performing metabolic stuff as enzymes in their own right, or whether they're just building peptides. And if it is possible, you know, if the RNA world hypothesis fits into your one, basically.
It could be. I mean, this is where it's like we ask the chemistry community and the biochem community for good ideas and good experiments to try. Because it's quite plausible, like that cycling system I showed you and that I showed you on the phone, in those random sequences probably are functional ribozymes.
In which case you could have metabolism without proteins to an extent of the
you could have the beginning of a proto-metabolism. Because the whole thing is based on this idea of scaffolding. So scaffolds are a structure that holds you up while more sophisticated things get built, like the scaffolding from a building or a church cathedral or something being done. You first have that scaffolding, and then you can get stuff to sort of start emerging. And when the keystone arch is put in, you know, in a great cathedral construction, they can take all the scaffolding away. And then you have a wonderful cathedral. And that's what we're kind of seeing in the cycling pools, this massive scaffold upon which the function of life can emerge. Thanks for coming in. Good to see you again. Be in touch. Yeah.
So we've had a lovely about hour and 15 minutes that we've all been sitting. I'm sure everyone might want to get around and get up and stretch a little bit. So maybe let's have a little bit of open discussion. So if you want to do small groups or you want to approach groups one-on-one, whatever, feel free. We'll get started on dinner prep, and then in 15-20 minutes, we'll start the next talk.
And it'll be shorter.